Welcome to the deep dive. Have you ever thought about the brain's immune system as, this dynamic, shape-shifting police force?
Most people haven't, right? I mean, we usually picture immune cells as just these static little Pac-Men floating around waiting to bump into something bad.
Exactly. But what if they actually deploy reserve troops based on, their physical distance from the danger? That's a totally different way of looking at biology.
It really is. And today we're unpacking a massive, just a huge aha moment in Alzheimer's disease research.
Yeah, this is coming out of a cutting-edge 2025 paper in Nature Neuroscience.
Right, by Alberto Ordura-Fabregat and a whole team of researchers.
And the title is a mouthful. It's called Response of Spatially Defined Microglia States with Distinct Chromatin Accessibility in a Mouse Model of Alzheimer's Disease.
Which sounds incredibly dense, but the core of it is just fascinating.
So our mission today is to look past the obvious stuff and really focus on the secret dynamic lives of these brain cells called microglia.
Because targeting the so-called bystander cells might actually be the key to fighting neurodegeneration.
Okay, let's unpack this. We are focusing entirely on microglia today. And these aren't your standard immune cells, right?
No, not at all. They have a very unique biological backstory. Microglia are the tissue-resident macrophages of the central nervous system.
Meaning they're the brain's first responders.
Exactly.
Yeah.
But unlike the rest of your immune system, which, comes from your bone marrow, these guys originate from embryonic yolk sac progenitors.
Wait, embryonic? So they get there before you're even born?
Yeah, they migrate into the developing brain incredibly early, like way before the blood-brain barrier fully closes.
Wow.
And once that barrier seals, they're essentially locked in for good. They maintain their own local population for your entire life.
They're the ultimate isolated locals. They can't just, call for massive backup from the bloodstream when things go wrong.
Right. They have to handle the neighborhood with the troops they already have.
And to study how these local troops handle a massive threat like Alzheimer's, the researchers used a very specific tool, the 5x-FAD mouse model.
Yeah, the 5-FAD model. So this is a genetically modified mouse that carries 5 human familial Alzheimer's mutations.
So it's basically programmed to get severe Alzheimer's pathology really fast.
Exactly. It triggers this rapid, highly aggressive overproduction of amyloid beta, which is the protein that forms the toxic plaques in Alzheimer's.
It gives them a hyper-accelerated window to study this stuff. So when they looked at the brains of these 5x-FAD mice, what did they see?
Well, they noticed a complete breakdown in the normal architecture of the microglia.
Because in a healthy brain, they're pretty organized.
Oh, yeah. In a healthy wild-type mouse, microglia are evenly distributed. They form this perfectly spaced grid.
Just keeping an eye on things.
Right. Every cell has its own territory. But under the stress of massive amyloid deposition, that grid just collapses. It forms what they call an aberrant bimodal distribution.
Bimodal, meaning it splits into 2 distinct groups.
Yes. Based entirely on their spatial location relative to the plaques.
And they could see this using a fluorescent dye, right? MethoxyXO4.
Yeah, methoxyXO4 binds specifically to the amyloid deposits. So it basically lights up the microscopic battlefield.
Okay, so what's the first group of microglia?
The first faction is PAM, which stands for plaque-associated microglia.
And these are the ones right in the thick of it.
Exactly. They are in direct physical contact with methoxyXO4 labeled amyloid, or at least within a tiny 30 micrometer radius.
And then there's the second group.
The non-PAM. These are located far away from the plaques, sitting out in the relatively healthy tissue.
And they look normal.
Totally normal. Highly ramified, deep branches, seemingly just doing their standard patrol.
Let's give you guys a mental image for this. Think of the amyloid plaque as a raging house fire.
That's a great analogy.
The Pan-Am are the firefighters physically spraying water on the flames, and the non-PAM are the patrol cars circling the neighborhood blocks away.
And for the longest time, the assumption was if you aren't at the fire, you aren't fighting the fire.
You're just a bystander. But the researchers used something called BRDU to see what these cells were actually up to.
Right. So BRDU is a synthetic building block of DNA. When you give it to an animal, it gets incorporated into the DNA of any cell that is actively dividing.
So it's a tracker for cell multiplication.
Exactly. Unsurprisingly, the PAM at the plaques had high BRDU uptake. They were proliferating.
Which makes sense. Big fire, you need more firefighters.
But the absolute shocker was the data from the non-PAM.
The ones blocks away.
Yes. They also showed a highly elevated proliferative capacity. They were multiplying just as fast as the PAM.
Okay, so just to recap this first part real quick for you listening, Brain immune cells in Alzheimer's split into two camps, PAM at the plaque and non-PAM far away. But both are highly active.
Right, which totally breaks the old bystander theory.
Which leaves us with a massive question. Why would the cells far away from the plaque be multiplying just as fast as the ones touching the damage? Like, what are they preparing for?
Well, answering that required tracking these distant cells over time to see where they ended up.
If they're far away, what is their actual job?
Right. And to do that, they had to use some incredibly ingenious genetic engineering.
The confetti mice. I love this part.
It's so cool. They used CX3, CRE1, CRE2, R26 confetti mice, and later TIMM119 CRE2 mice.
Okay, that is a lot of letters and numbers. Break down the confetti mouse for us. How's that actually work? You can't just paint a cell and hope the paint stays on.
No, you can't. It relies on a genetic trick called the CRE-lock system.
Okay.
Basically, they engineer the mice so their microglia carry an enzyme called CRE recombinase.
But the enzyme is locked away, right?
Yes. It's locked out of the nucleus. It can't touch the DNA until the researchers give the mouse a specific drug called tamoxifen.
So tamoxifen is the key that unlocks the door.
Exactly. Once unlocked, the CRE enzyme gets into the nucleus and acts like molecular scissors.
Snip.
It cuts and splices the DNA in a way that randomly locks the cell into expressing one of four bright fluorescent colors.
Nuclear green, cytoplasmic yellow, cytoplasmic red, or membrane cyan.
Right. And once that DNA is cut, it's permanent. That cell and every daughter cell it ever makes will glow that exact color.
It's like putting different colored, permanently glowing sports jerseys on random people in a massive crowd so you can track exactly where they run over the course of the game.
That's exactly it. But they had to be careful. They only wanted to put the jerseys on the non-PAM patrol cars.
Right, because if you tag everybody, you don't know who started where.
Exactly. So they needed specific protein markers to tell them apart before they moved.
And what did they find?
They found that a marker called CDL1C is almost exclusively on the PAM. The firefighters.
And what about the patrol cars?
The non-PAM heavily express a marker called TMEM119.
So TMEM119 is the patrol car badge.
Right. So they use that TMEM119 marker to drive the confetti color change.
Yeah.
Only the distant cells got tagged.
And then they just, waited?
Yeah, they let the clock run for 2 to 8 weeks.
And the discovery here is wild. Where did those colored cells show up?
They showed up right next to the methoxy XO4 labeled plaques.
As PAM.
Yes. What's fascinating here is that they didn't just migrate. They physically transformed. They lost the TMEM119 badge and gained the CD11C uniform.
Okay, quick recap on this. Using this fluorescent confetti tracking, we learned that distant TMEM119 positive non-PAM physically transform into CD11C positive PAM at the plaque site.
For a period of 2 to 8 weeks, yes.
So think about that. If you were designing a drug, would you want to target the cells already at the fire or the vast reserves waiting in the wings?
It really shifts the therapeutic focus.
Yeah.
Because the non-CAM aren't bystanders. They are the reserve troops.
But here's where it gets really interesting. When they arrive at the plaque, they don't just sit there and hold a hose.
No, they don't. They undergo what's called clonal expansion.
Meaning they clone themselves.
Exactly. A non-PAM arrives, senses the amyloid threat, and starts rapidly copying itself to build a wall of identical clones.
And they could see this because of the colors, right? An entire cluster of red cells surrounding a plaque.
Yes, visually it looks like a clone. But in science, you have to prove it mathematically.
You can't just eyeball it and say, yep, that looks red.
Right. So they used a Monte Carlo simulation.
Oh, I love this part. Monte Carlo, explain that.
It's a computer algorithm. It basically generates thousands of random cell distributions in a 3D space to see what pure chance looks like.
To make sure all the red cells didn't just accidentally wander into the same spot.
Exactly. And the statistical proof was undeniable. The clustering of same-colored cells wasn't random. They were actively cloning themselves at the plaque site.
So they're building these localized walls to restrict the amyloid.
Yes. But then they needed to measure how big these walls were getting.
How do you measure an irregular 3D microscopic wall of cells?
By building Voronoi grids.
Voronoi grids. Okay, break that down for me.
Imagine plotting the center of every colored cell. A Voronoi diagram draws geometric borders around each point.
Like drawing a bubble around each cell.
Right. So every point inside a specific polygon is closer to that cell's center than any other cell. It lets you calculate the exact 3D volume of the clone.
And then they compared the size of the microglial clone to the size of the amyloid plaque.
Yes. And initially, it correlated perfectly. Bigger plaque, bigger clone.
The immune system was scaling its response.
But then they hit the bad news.
The tipping point.
Yes, they found a hard limit. Once an amyloid plaque exceeded a volume of 1,000 cubic micrometers,
The correlation just stopped.
it completely uncoupled. The plaque kept growing, but the microglia clones hit a wall. They couldn't expand any further.
The amyloid deposition literally outcompetes the cloning rate of the microglia. The fire just gets too big for the firefighters to surround.
The immune system essentially fails.
Okay, third recap here. PAM clone themselves to build walls around plaques. But once a plaque hits 1,000 cubic micrometers, the microglia max out, and the plaque grows unchecked.
It's a devastating biological limit.
So, knowing there is a physical limit to the natural immune response, how can we chemically boost that 1,000 cubic micrometer ceiling? Because so far, the immune system eventually loses the race.
Right.
What does this all mean for finding an actual treatment? Did they find a way to intervene?
They absolutely did. They had to find the biochemical switch that regulates that transition from non-PAM to PAM.
The dispatch signal.
Exactly. And through transcriptomic analysis, they found it. It's a signaling pathway driven by CSF1.
Colony stimulating factor 1.
Yes. CSF1 is a ligand, a molecule that binds to the CSF1R receptor on the surface of the microglia.
So when CSF1 binds, it's basically the radio dispatch telling the non-PAM to get moving, transition, and start cloning.
You got it. So the researchers treated the 5x FAD mice directly with CSF1.
They flooded the system with the dispatch signal, and what were the results?
First, it significantly reduced the overall amyloid plaque volume.
Huge.
But the mechanism was the real victory. Remember the Voronoi grids?
Yeah, the volume measurements.
Under CSF1 treatment, the correlation between plaque size and clone size was much tighter. And crucially,
What about the massive plaques, the ones over 1,000 cubic micrometers?
the ceiling was broken.
Wait, really?
Yes. The PAM clones were finally able to keep expanding past that 1,000 micrometer limit. They matched and restricted even the most massive amyloid deposits.
That is incredible. They chemically removed the biological limiter.
They did. But, biology is noisy. How do we know it was specifically CSF1 and not just general immune system panic?
Right, you need a negative control.
Exactly. So they tested another ligand called IL-34.
IL-34.
Yeah. It's structurally very similar to CSF1, and it binds to the exact same CSF1R receptor on the microglia.
So two different keys fitting into the exact same lock.
Right. So you might expect it to do the same thing. But IL-34 did absolutely nothing.
Nothing.
Nothing. It didn't boost the non-PM transition. It didn't increase clonal expansion, and it didn't reduce the plaque size.
Wow. So quick recap on the intervention. CSN signaling is the master switch that boosts the non-PAM to PAM transition, allowing the immune system to surround even the biggest plaques.
While a structurally related molecule like IL-34 does zero.
So think about how precise biology is, Why does CSF1 work miracles here while IL-34 does nothing? It shows you have to have the exact right key.
It really does. And if we connect this to the bigger picture, it completely reframes Alzheimer's therapeutics.
How so?
Well, for decades, billions of dollars have been spent trying to create drugs that attack or clear the amyloid plaque directly.
Just blunt force plaque removal.
Right. But this study proves that our brain already has a dynamic, shape-shifting reserve army. The non-PAM.
The patrol cars waiting in the wings.
The key might not be attacking the plaque itself, but giving those non-PAM the biological signals like CSF1 to enter the fray before the plaque outcompetes them.
So you, listening to this, you now understand the spatial dynamics of the brain's immune system way better than most people.
You know that cells aren't static. They respond, they migrate, they change identity based on distance.
Exactly. But I want to leave you with one final kind of provocative thought.
Go about the environmental cues.
Yes. Deep in the paper, they briefly mention that these complex behaviors, like when a non-PAM decides to transform, can be altered by outside factors.
Things like systemic inflammation, early antibiotic use, even the gut microbiota.
The gut-brain axis. So if these non-PAM reserve troops are constantly listening to their environment to decide when to activate,
how might your daily lifestyle, your diet, or the health of your gut be whispering commands to the immune cells deep inside your brain right now?
Are your daily habits sending the right dispatch signals? It's something to really mull over. Keep diving deep.